Hi students, we are going to discuss today about the oligosaccharides, lignans, glucosinolates. Oligosaccharides, lignans, glucosinolates, most of these molecules are the secondary metabolites and part of the plant system. Oligosaccharides, lignans, glucosinolates earlier have shown as some of them as anti-nutrients, but the current research have shown the health benefits of these important molecules in the human system, particularly oligosaccharides and lignans, glucosinolates in various ways, like working either for antioxidant activity or against the cancer or the cardiovascular diseases. A few examples like stachyose, verbascose, these are the molecules in the oligosaccharides group, and lignans which we have from the various plant systems like flax seeds, and glucosinolates an example of from mustard, cabbage, etc. works against the free radicals or the cardiovascular diseases, or for that matter even in cancer too. Oligosaccharide is a carbohydrate polymers comprise 3 to 10 monosaccharides or simple sugars. They were linked together by glycosidic bond through condensation reaction between an anomeric carbon of monosaccharide and the other one. They can also form N-glycosidic linkages under certain atmosphere. The minimum number of reducing sugar components is one molecule lesser than the total number of simple sugars. Reducing sugar can be characterized from the hydroxyl group, that is OH group, on the anomeric carbon. What are the food sources for oligosaccharides? Oligosaccharides are one of the components of fiber found in plants. Fructose, oligosaccharides and inulin are found naturally in Jerusalem artichoke, chicory, onions and asparagus. Fructose oligosaccharides products derived from chicory root contains significant quantities of inulin, a fiber widely distributed in fruits, vegetables and plants. Health benefits of functional oligosaccharides. Functional oligosaccharides have been applied for many purposes such as nutrients, pharmaceuticals, feeds, cosmetics, immunostimulating agents and prebiotic compounds, which incorporate 13 classes of 58 commercially produced non-digestible oligosaccharides showing bifidogenic functions. In addition, known functional oligosaccharides also include arabino-oligosaccharides, arabino-galacto-oligosaccharides, arabino-xylo-oligosaccharides, galacto-urano-oligosaccharides, ramno-galacto-urano-oligosaccharides, and human milk oligosaccharides. Like there are various oligosaccharides and having a distinct and different functional characteristics for these oligosaccharides. Cyclodextrin alpha beta gamma are cyclic alpha 1,4 glucans with degrees of polymerization of 6, 7 and 8 monosaccharide units respectively. Macrocyclic carbohydrates have been widely applied as building blocks in supramolecular chemistry, drug carriers, molecular reactors and artificial receptors. The use of functional oligosaccharides improves the balance of intestinal microflora and greatly decreases the gastrointestinal infections. Additionally, the consumption of functional oligosaccharides can reduce the risk of lifestyle related diseases such as cardiovascular diseases, cancer, obesity and type 2 diabetes, which are related to obesity. Thus, functional oligosaccharides are widely cited to be important dietary fibers in nutritional advice for metabolic syndromes induced specific disorders. So, various examples of oligosaccharides: arabinoxylon oligosaccharides derived from the cereal grains. Fructo-oligosaccharides, they are called FOS or oligofructose, Jerusalem artichokes, onions, canned foods are the examples for FOS. GOS, that is Galacto-oligosaccharides, the molecules are raffinose, stachyose and verbascose. They are widely present in beans, peas, lentils, cabbage, whole grains, soybean oligosaccharides in soy, and trans-galacto-oligosaccharides. Gentio-oligosaccharides produced from postulon, gluco-oligosaccharides produces from sucrose, human milk oligosaccharides from the human breast milk, isomalto-oligosaccharides or IOS produced from the starch, lactose-sucrose produced from the lactose and sucrose, maltotriose produced from the starch during digestion, found in liquid glucose, brown rice syrup, mannan-oligosaccharides or MOS artificially produced, melibose-derived oligosaccharides, N-acetyl-keto-oligosaccharides derived from the chitosan, pectic oligosaccharides, they are derived from the pectin actually, xylo-oligosaccharides produces from the corn cob and birchwood is an example for xylo-oligosaccharides. Oligosaccharides are often added to commercial foods as sweeteners or as fiber even. If we look into the digestion, fermentation and absorption function and the side effects of these oligosaccharides, except maltotriose are indigestible, which means human lacks enzyme to break them down in the small intestine. So they reach the large intestine where beneficial colonic bacteria break them down, ferment to absorbable nutrients which provide some energy, about 2 calories per gram in average. Certain breakdown products of oligosaccharides, namely short chain fatty acids, may have beneficial effects on large intestinal lining. Most oligosaccharides act as a soluble fiber which may help prevent constipation. Ingestion of large amount of oligosaccharides can result in abdominal bloating and excessive gas, that is flatulence. Other uses of galacto-oligosaccharides: number of oligosaccharide molecules may form polysaccharides through multiple linkages between the anomeric carbon at the end of a molecule and the hydroxyl groups of another oligosaccharide molecule. Through glycosidic linkage and N-glycosidic bond, oligosaccharide may react with lipids and form lipopolysaccharides and saccharolipids. N-linked oligosaccharides can also react with side chains of amino acid residues, particularly asparagine from a protein, to form a glycoprotein. Oligosaccharides are known to be found in glycolipids and glycoproteins. Some of them are found from the breakdown of starch and cellulose. They are called cellodextrin and maltodextrin. Chemical making is one of the functions of oligosaccharides. This is because they have much variation, similarities. For instance, blood types are marked by oligosaccharide. A blood type has one oligosaccharide, B blood type has one too. Both of these oligosaccharide markers are present in AB blood type while O blood type has none. The reason why blood needs to be typed before transfusion is because these oligosaccharides in blood are different enough than each other to be attacked by the body's immune system. Oligosaccharides can be identified as antigens defected by immune systems with incompatible blood as foreign pathogens. If transfusion were to occur with incompatible blood types, clotting and major illness would occur, ultimately causing death. However, type AB blood contains all the possible oligosaccharides, combinations possible A or B, and since type O blood has no markers attached to it, type AB blood carriers are generally called universal acceptors. Whereas type O blood carriers can only accept blood transfusion from other type O blood donors which do not contain any oligosaccharides present within red blood cells. Oligosaccharides as prebiotics. Prebiotics are digestible nutrients that selectively promote the growth of normal intestinal bacteria that may have beneficial effects on the large intestinal lining. Oligosaccharides currently considered as prebiotics include fructo-oligosaccharides, FOS or oligofructo, and trans-galacto-oligosaccharides. You should be very clear in understanding of these prebiotics and probiotics, which we have discussed in our earlier class about the curd as a probiotic which has microbiome. Whereas in prebiotics, these fructo-oligosaccharides is an example and which also mimic and they can also promote the growth of normal intestinal bacteria. So galacto-oligosaccharides are synthesized through an enzymatic conversion of lactose. It is comprised of chain units of galactose group through consecutive trans-galactosylation reactions, which the degree of polymerization ranging from 2 to 8 monomeric units. It is known that these new class of prebiotics have an important role in improving gut health by sustaining beneficial and balanced gut microbiota. Today, many infant formula companies have galacto-oligosaccharides in their formula milk to improve the gut health. Human milk oligosaccharides. Human milk oligosaccharides are complex glycans that can be found in breast milk. One of the most important factors in infants' diet is from breast milk, which pertains one of the most complex group of oligosaccharides known as human milk oligosaccharides. They are found in 3, 4, 5 or even 6 chain sugars. For examples, some of the human milk oligosaccharides include raffinose, 2-fucosyl lactose, 3'-fucosyl lactose, 3'-sialyl lactose, 6'-sialyl lactose, and lactose and tetrose. These human milk oligosaccharides differ in their size, structure, and specific linkages. There are more than 150 distinct human milk oligosaccharide structures out there that are identified so far. All these human milk oligosaccharides are distinct in their structure, acidity, and functions too. The backbone of human milk oligosaccharides is the disaccharide lactose, which is formed by the linkage between galactose and glucose sugars. The final structure of human milk oligosaccharides all depends on whether the backbone lactose is fucosylated or sialylated in either beta or alpha configurations or at different carbons. For example, 2'-fucosyl lactose has fucose group at the alpha-1,3 position of the glucose monosaccharide of the lactose. Being sialylated means the addition of sialic acid group and formation of an acidic human milk oligosaccharide. When the protein alpha-lactalbumin, also referred as LA-LBA, is present, the enzyme beta-1,4-galactosyl transferase changes its function. It connects galactose to glucose sugars which form lactose. Human milk oligosaccharides are formed from lactose sugars, but the exact mechanism for this transformation is still unknown. Human milk oligosaccharides are produced only when woman is lactating and are formed in the mammary gland. Then we discuss a little bit about the lignans. Lignans are a type of plant compounds known as polyphenols. These comes under the group of polyphenols. Lignan precursors are most abundant in flaxseed, although they are also found in other seeds like sesame seeds, berries, fruits, vegetables, and whole grains are also present these lignans. Plant lignans are polyphenolic substances derived from the phenylalanine via dimerization of substituted cinnamic alcohols known as monolignals to a dibenzyl butane skeleton. This reaction is catalyzed by oxidative enzymes and is often controlled by dirigent proteins. What are the food sources for these lignans? Plant lignans are co-passengers of dietary fiber, and therefore fiber-rich food items are often food good sources of lignans. Flaxseed and sesame seed contain higher levels of lignans than most other foods. The principal lignan precursor found in flaxseed is seco-isolaricinol diglucoside. Other sources of lignans include cereals, that is rye, wheat, oat, and barley, rye being the richest source, soybean, cruciferous vegetables such as broccoli and cabbage, and some fruits, particularly apricots and strawberries, are the rich sources for these lignans. Seco-isolaricinol and metiracinol were the first plant lignans identified in foods. Pinoresinol and laricericinol are most recently identified plant lignans that contribute substantially to the total dietary lignan intakes. Typically, laricericinol and pinoresinol contribute about 75% to the total lignan intake, whereas seco-isolaricinol and metiracinol contribute only about 25%. The distribution may change as the distribution of syringericinol and hydroxymedricinol have not properly been quantified in foods. When a part of the human diet, some plant lignans are metabolized by intestinal bacteria to mammalian lignans, enterodiol and enterolactone. Lignans that can be metabolized to mammalian lignans are pinoresinol, lariciresinol, secoisolariciresinol, metyrresinol, hydroxymetyrresinol, syringerasresinol, and sesamin. Lignans are one of the major classes of phytoestrogens, which are estrogen-like chemicals and also act as antioxidants. The other class of phytoestrogens are isoflavones and comistans. Health benefits of lignans. Many studies have revealed in the cardiovascular diseases, diets rich in foods containing plant lignans, whole grains, nuts, seeds, legumes, fruits, and vegetables have been consistently associated with the reductions in the risk of cardiovascular disease. It is likely that numerous nutrients and phytochemicals found in these foods contribute to the cardioprotective effects. Maybe it is the overall effect, but people have shown in isolated cases also, like using the plant lignans, also have shown wonderful effects against the cardiovascular diseases. Ovarian and uterine health. Lignans have anti-estrogenic properties that mimic estrogen and bind to estrogen receptors, but they do not have the same antagonistic effects of estrogen. Actually, that's the beauty of these plant lignans. Plant estrogens are a cleaner form of estrogen, and when more plant lignans are consumed, more normally occurring endogenous estrogens is flushed out of the body. It is believed that this is the mechanism behind the studies that indicate better ovarian and uterine health. Prostate health. Study found that the vegetarian men, particularly the people who are eating only plant foods, and particularly with the diets high in plant lignans, like the people who are taking more of the whole grains, nuts, seeds and legumes and flax seeds, etc., have better prostate health than omnivorous males. In the sense, the people who consume both vegetarian and non-vegetarian diet, if you compare, the vegetarians who are taking lots of lignans have better prostate health. Plant lignans give rise to the mammalian lignans, enterodiol and enterolactone. The richest source is linseed. Colon health. Flaxseed, the richest source of mammalian lignan precursors such as secoisolarcisenol diglycoside, has been shown over the short term to decrease some early markers of colon health risk. Flaxseed hulls are high in fiber and lignans. The fiber is good for digestion, absorbing impurities, and cleaning the colon. Fiber has been known to improve bowel movements as well as reduce the rate of colon cancer. Hot flashes and menopause, where a Mayo Clinic study shows that lignans provide a more natural estrogen treatment for menopause and hot flashes than synthetic hormone therapy, and therefore are potentially healthier treatments for both. Even soybean has been shown particularly for this menopause and hot flashes, where it also contains like genistein, diadygin, other compounds are also beneficial. Hair health. There are studies that indicate that people with high amounts of lignans in their bodies have better hair health. Poor hair health can be caused by hormone DHT. Flaxseed lignans can inhibit the production of enzyme that converts testosterone into DHT. Now we discuss about the glucosinolates. The glucosinolates are natural components of many pungent plants such as mustard, cabbage, and horseradish. The pungency of those plants is due to mustard oil produced from glucosinolates when the plant material is chewed, cut, or otherwise damaged. These natural chemicals most likely contribute to plant defense against pests and diseases, but are also enjoyed in small amounts by humans and are believed to contribute to the health promoting properties of cruciferous vegetables. If we look at the chemistry of these glucosinolates, constitute a natural class of organic compounds that contain sulfur and nitrogen and are derived from the glucose and an amino acid. They are water-soluble anions and can be leached into the water during cooking. Glucosinolates belong to the glucosides. Every glucosinolate contains a central carbon atom which is bound via sulfur atom to the thioglucose group and via a nitrogen atom to a sulfate group, making a sulfate aldoxine. In addition, the central carbon is bound to a side group. Different glucosinolates have different side groups, and it is variation in the side group that is responsible for the variation in the biological activities of these plant compounds. Some glucosinolate, sinigrin, is the precursor to allyl isothiocyanate. Glucotropylene is the precursor to benzyl isothiocyanate. Gluconastatolylene is the precursor of phenethyl isothiocyanate. Glucoraphanin is the precursor to sulforaphane. These are the precursors of these molecules. So now we look into the plants with glucosinolates. Glucosinolates occur as secondary metabolites of almost all plants of the order Brassicales. Example, families of Brassicaceae, Cruciferae, Capardicaceae and Caricaceae, but also in the genus Drypetes, family Putragiaceae. For example, glucosinolates occur in cabbages, white cabbage, Chinese cabbage, broccoli, watercress, horseradish, capers and radishes. They are typically in pots consumed with the pungent taste of these vegetables due to breakdown products, isothiocyanates or mustard oils of glucosinolates. The glucosinolates are also found in the seeds of these plants. If we look at the biochemistry and the enzyme activation, about 132 different glucosinolates are known to occur naturally in plants. They are synthesized from certain amino acids, so-called aliphatic glucosinolates, derived from mainly methionine, but also alanine, leucine, isoleucine or valine of various amino acids. Most glucosinolates are actually derived from chain elongated homologous of these amino acids. Example, glucoraphanin is derived from the dihomomethionine, which is methionine chain elongated twice. Aromatic glucosinolates include indolic glucosinolates such as glucobrassicin, derived from tryptophan, and others from phenylalanine, its chain elongated homologous homophenylalanine, and cinalbin derived from tyrosine. Enzyme activation. The plants contain the enzyme myrosinase, which is in the presence of water cleaves off the glucose group from a glucosinolate. The remaining molecule then quickly converts into a isothiocyanate, a nitrile or a thiocyanate. These are the active substances that serve as defenses for the plants. Glucosinolates are called mustard oil glycosides. The standard product of the reaction is the isothiocyanate. The other two products mainly occur in the presence of specialized plant proteins that alter the outcome of the reaction. To prevent damage to the plant itself, the myrosinase and glucosinolates are stored in separate compartments of the cell and come together only or mainly under the conditions of physical injury. The biological effects, if we look at toxicity levels also, the use of glucosinolate containing crops as primary food source for animals can have negative effects if the concentration of glucosinolate is higher than what is acceptable for the animal is questioned. Some glucosinolates have been shown to have toxic effects, mainly as goitrogens, in both humans as well as in animals at higher doses. However, tolerance levels to glucosinolates varies even within the same genus. Example, Ecomys cacrinus and Ecomys rosatus. Taste and the eating behavior. The glucosinolate sinigrin among others was shown to be responsible for the bitterness of the cooked cauliflower and Brussels sprouts. Glucosinolates have been shown to alter animal eating behavior. Many studies have been conducted on the plants producing large amounts of glucosinolates under potential action against cancer with the hydrolysis product of glucoraphanin. Most recently, a randomized control study evaluating the effects of broccoli seed supplementation on autism spectrum disorder is underway. The makeup of glucosinolates and their hydrolysis products varies by vegetable. insects, substances derived from plants producing large amounts of glucosinolates can serve as natural pesticides. The large white butterfly oviposes its eggs on these glucosinolate containing plants because they help the larva survive. In contrast, the diamond block moth possesses a completely different protein, glucosinolate sulfatase, which desulfates glucosinolates, thereby making them unfit for the degradation to toxic products by myrosinase. Other kinds of insects, specialized sawflies and aphids, sequester of glucosinolates. This diverse panel of biochemical solution to the same plant chemical plays a key role in the current attempts to understand the evolution of plant-insect relationship. It has a powerful antioxidant activity. Like vitamin C, vitamin E and carotenoids are direct antioxidants as they neutralize free radicals before they can harm cells. Glucosinolates and their hydrolysis products are considered as indirect antioxidants as they do not neutralize free radicals directly but rather by modulating the activity of xenobiotic metabolizing enzymes, that is phase 1 and phase 2 enzymes that trigger the longer lasting antioxidant activity. Phase 1 enzymes generally increases the reactivity, and phase 2 enzymes, particularly glutathione transferase, aldehyde reductase, etc., increases the water solubility. Hence, inhibition of phase 1 and induction of phase 2 enzymes are necessary for the protection of cells against DNA damage by carcinogens and reactive oxygen species. The genes for the phase 2 enzymes contain a specific sequence of DNA called antioxidant response element. These GSL hydrolysis products works as an indirect antioxidant, works effectively against the free radicals. Hence, there is a compelling evidence that GSL can help reducing the damaging effects of free radicals both directly as well as indirectly. Students, now what we have learned so far in this class about the oligosaccharides, lignans and glucosinolates. In conclusion, what we have learned that as a prebiotics, particularly the oligosaccharides, their importance in the human milk, lignans are useful in dietary management of cardiovascular disease and neural diseases, with flaxseed as a dietary source, particularly for lignans. Glucosinolates explains their importance in maintaining the immune system, destroying the free radicals produced during oxidation. Thus, these three important molecules from the various food sources are better sources in augmenting or to prevent the cardiovascular diseases, cancer, diabetes, and improves the gut health effectively. Thank you.